Once nutrients diffuse or move towards the surface of the root, it's then that they're absorbed by the plant. And nutrient absorption is the most important part of the cycling of nutrients after supply. Now, understanding how much nutrient is absorbed by a given plant is a pretty simple function. If you know the supply of nutrients in soils, the length of roots in the soil, and the activity of a given unit of length, you can calculate total uptake. Now, of these three, supply rate is the major factor that determines uptake. And for reasons that we'll talk about, root length and the absorption properties of roots in many circumstances are less important. The authors state that across a broad range of nutrient availability, nutrient absorption by vegetation is driven primarily by nutrient supply. So if we have supply times length times activity, for some reason length and activity must be less important such that supply correlates better with uptake. In general, it's important to know that root length is a better predictor of nutrient absorption than is root biomass or root surface area. And the reason for this, in part, is that if you have a given root and it's relatively thick or it's relatively thin, there's still a diffusion shell that surrounds these roots. So if it's thick or not, that doesn't really affect what the size of that diffusion shell is. And thinking about it in other terms, if this root is really dense and it has a lot of biomass compared to another root, that doesn't necessarily increase the amount of nutrient that can be taken up. In the same sense, if you can imagine a root that was built like this versus one that had a lot more surface area, and it looks something like that, which roots never do, but just imagine that they did, the diffusion shell that would be generated around each of those would still be pretty similar, because diffusion is more important than surface area in generating a concentration gradient. So root length is really the better predictor of nutrient absorption than is root biomass. In this figure, figure 8.4, this is model synthesis. And what it is it takes a look at the effect of changing parameter values in the model that simulates phosphate absorption. Now think about phosphate as being a relatively non-mobile nutrient by roots. And we can take a look at the relative importance of changing different factors by standard amounts. So we standardize all the parameters to 1 and take a look at what the effect is of doubling that parameter. And that's what the x-axis is here, change in parameter. And y looks at the change in phosphorus absorption, micromoles per pot. So if you have a plant growing in a pot, and you can change a bunch of different things for that plant, what are the effects of changing how fast roots elongate, for example, versus transpiration rate? And in a model, those factors that are up here, a doubling of that factor has a much larger effect than those factors that are down here. So which factors don't have a big effect? Something like transpiration rate. And we talked about how mass flow just isn't that important compared to diffusion, which is just a lot faster. So changes in transpiration rate really don't pull a lot more nutrients to the plant. Also, this is called the maximum absorption rate. It's an Imax. And when people model the uptake of nutrients and they take a look at the concentration of the nutrients in the soil solution, and uptake is here, there's usually a minimum concentration that's required for uptake, and then it has a functional response that looks like this. And that maximum uptake rate is shown here. So what happens then if you double the maximum uptake rate to something like that? So at a given concentration, there's a plant that can take up twice the rate of nutrients. Shouldn't that lead to greater P absorption? It would if the concentrations were high next to the root, but generally the concentrations next to the root are relatively low. And so doubling the maximum uptake rate doesn't actually increase the uptake rate much, because most of the concentrations next to the root are relatively low, close actually to the minimum concentration. So changes in transpiration rate don't help much. Changes in maximum absorption rate don't help much. And then you have two different things in the middle, and it's really, in this case, root elongation rate and soil P concentrations that really determine most of the phosphorus absorption for this situation where you had a plant growing in a pot. Now, we talked about whether root elongation, what that actually does. And in a situation where you have a young plant in a pot, root elongation allows plants to explore more of the soil. And so this is really an index of the ability to explore the soil. Most important really are these factors here, is thinking about what's not important. And transpiration rate and maximum absorption rate are not. Total root length is, but up to a certain point. And we're going to talk about when root length doesn't become important. For all intents and purposes, after a certain point, root length is just important for competition between roots. And I'll explain that a little bit here. So these are the root systems of two different plants. On the left, we have a forb. These are both grassland plants. This is a forb, a wildflower called Solodega rigida, and this is stiff goldenrod. And this is Schizocarium scoparium. It's a little blue stem. And you can see, for all intents and purposes, if you grow the plant the same amount of time, the root systems aren't that different. Looks like this one has a little bit more roots, but they're both about the same depth and they take up most of the soil profile. This distance right here is on the order of 1 meter from top to bottom. So they really fill up the soil profile to the same degree. But if you grow these two together, this one, for all intents and purposes, loses out and is quickly extirpated from that soil. And instead, it's this species that wins. So why would that be? Why would this grass be able to outcompete this plant based on what we know of nutrient uptake patterns? Well, when we take a look at what happens in roots, taking up nutrients, we talked about how there's depletion zones around the roots, and you get really low concentrations next to the roots and high concentrations away from the roots. So if we had a situation where you had two different plants growing in soils, and this could be species A with the black roots and species B with the white roots, that the nutrient supply in that soil gets partitioned among those species. The consequence of all of this root length in here doesn't necessarily lead to greater nutrient uptake after a certain point. You can see that all the nutrients that are made available in the soil are going to diffuse to one of these species, either the black one or the white one, depending on where it is in the soil and how much root is present from one species to another. What happens if we start to add more roots? So we can partition, in this case right here, nutrient supply is either going to the black root or the white root, and if the white species adds more root length, it just increases the proportion of the supply that it acquires. It doesn't actually increase the total uptake from the two plants. So adding additional root length just increases the fraction of the supply that you get, not the total supply that's acquired by the two plants. So when we take a look at plants and we look at all the roots that are there, a lot of that root length is there just for competitive purposes. There's way more roots that are present in soil than is needed to take up the nutrient supply. After a certain point, those roots are there to make sure that competitors don't get those nutrients and the plants get it instead. In fact, when you take a look at a plant like this, they would be able to take up the same amount of nutrients with just 1% of the roots there. So if you get rid of 99% of the roots, they still can take up the same amount of nutrients. The other 99% of the roots that are there are just there for competitive purposes. So when we think about supply times length times activity, increasing the activity doesn't matter much because we saw in the model before that activity rates aren't that important because concentrations are really low at the root surface. And so being able to take up nutrients at a faster rate, you just still have to wait for nutrients to diffuse to the soil, to the root surface. So activity doesn't matter too much. Length at a certain point also doesn't matter too much. When plants are just starting to grow, length is really important, but additional length really is just redundant and doesn't increase the total uptake, but increases the fraction of the supply that an individual plant gets. So it's there for competitive purposes. So activity doesn't matter because concentrations are low. Length after a certain point doesn't matter because it's redundant. And so therefore, when we're taking a look at uptake, these two parts don't matter, and supply is the most important factor that determines uptake. In addition to roots taking up nutrients, also there's mycorrhizal fungi that take up nutrients and deliver those nutrients to the plants. Now, mycorrhizal fungi form symbiotic relationships between plant roots. Mycorrhizae is a specialized term, and what a mycorrhizae is a symbiotic relationship between plant roots and fungal hyphae. So we tend to talk about mycorrhizal fungi, or the type of fungus that form mycorrhizae, which is really the plant-fungus interface. So mycorrhizal fungi form these symbiotic relationships with plants. Why does that happen? Essentially, the mycorrhizal fungi serve as subcontractors, and the plants subcontract some of the activities to the mycorrhizal fungi. Why would they do this? Well, fungi have a few natural advantages over roots. First off, they're thin. And what that allows plants to do is, instead of producing one relatively thick root, they can produce a lot more length, or have access to a lot more length, because fungi are thinner than roots. So, for example, here's a photograph of a root. Here's individual root hairs, and here are mycorrhizal hyphae. So the fungus has penetrated the plant, and these are some of the hyphae that are coming off of the plant. Mycorrhizal fungi are really important for short distance resource acquisition. They don't form long hyphae necessarily, but are really important for, just like root hairs, for getting out slightly away from the root, outside of a depletion zone, and being able to acquire nutrients. They're really important for low diffusion nutrients. So in a high diffusion nutrient, you can have a root that goes into the soil, and it can sample a large volume of soil. But if that nutrient actually doesn't diffuse well, the depletion zone around the root is relatively narrow. And in that case, by having mycorrhizal hyphae that go out into the soil, you can extend that depletion zone for an individual root out further. Now, there are different types of mycorrhizal fungi, and this is a course of study unto itself. I'm going to go through just two of the differences right here. Some plants are non-mycorrhizal. They actually don't use mycorrhizal fungi at all. And then there are some that use or subcontract to arbuscular mycorrhizal fungi. And we'll often refer to these as arbuscular or AM. So arbuscular mycorrhizal fungi are important because they grow through the cell walls of the root cortex. And when you take a look at in the plants, they form these bodies, which are called arbuscules, which is where arbuscular comes from. And arbuscules are structures that exchange nutrients and carbohydrates. Where do you find AM, or arbuscular mycorrhizal fungi? Well, herbaceous communities, for example. Most of the plants in grasslands, for example, are arbuscular. So the grasses and the forbs use AM fungi, and a lot of herbaceous plants are arbuscular. In addition, in forests, it's generally the low phosphorus forests, tropical forests, for example, that really rely on AM more than the colder, more phosphorus rich forests. Now, in contrast to AM, there's also ectomycorrhizal fungi. So ecto is another shorthand for them, and they look pretty different. So the roots that form in plants that are colonized by ectomycorrhizal fungi are really relatively stubby. They're not long at all. And these are just major points of transport for nutrients between the mycorrhizal hyphae, which extend off from here and you don't see, and the root. So there's this really big interface. As far as the root is concerned, the ectomycorrhizal fungi don't penetrate the root. They form a net around that root, or the mantle as it's called. So you have hyphae that come out, there's a sheath around the root of exchange, and those are what ectos look like. Here's another example. Oh, sorry. Here's another example of an ectomycorrhizal plant, and you can see it just looks different. Really short, stubby roots. Now, ectomycorrhizal fungi are a stronger subcontract than our arbuscular mycorrhizal fungi, and ectos have unique abilities. For example, they can produce proteases, and those proteases make available amino acids by breaking down proteins that then diffuse and are taken up by the hyphae and then transferred to the plant. So having proteases in an organic soil where nitrogen is limiting is really important, and a lot of the ectomycorrhizal species that we know are found in these places with a lot of organic matter and nitrogen is limiting. So you go into low pH systems, you'll find arbuscular mycorrhizae. Go into high phosphorus, high organic soils where nitrogen is limiting, you tend to be more likely to find ectos because of the ability to access proteins directly.